right, so in this chapter, we're going to be focusing on the cell. Cell is the smallest unit of life, and we're going to talk about single-celled and multicellular organisms. We're going to talk about different kinds of cells, and we're going to talk about all the different cell parts and what they do. So, first of all, we didn't really know much about cells until the 1600s when Hooke, when I already circled here, Robert Hooke, he developed the first microscope. And when he developed the first microscope, he looked at cork, or like dead bark from a tree, and he saw, really all he really saw was the cell walls that were left behind because the cells were dead. But it reminded him of the rooms where the monks studied, and he called them cells. And the word cell sort of stuck, and we still use that word. Other scientists after him, Lewin Hooke, they mentioned here how he had better microscopes and he looked at different kinds of cells. And there were many other scientists along the way that have paved the way for everything that we know today about cells. Robert Brown, Schleiden and Schwann, etc. So this is sort of an overview. We're going to introduce the cell. We're going to talk about the nucleus and the ribosomes. We're going to talk about something called the endomembrane system. We're going to talk about the organelles that are specifically involved in energy. And then we're finishing up with the cytoskeleton, because yes, a cell actually has its own sort of skeleton. And we're going to finish up with that. So, introduction. So first of all, let's talk a little bit about microscopes. We're not going to spend a lot of time on that. We already covered the microscope earlier this year. But there are really three kinds of microscopes that they mention in this chapter, and a lot of the pictures you're going to see throughout the chapter were taken with these three different types of microscopes. So the first one is the light microscope. This is what we've used in the lab. We will use the light microscopes throughout the year. And it can display living cells. So that's a benefit of the light microscope, is we can actually look at like little structures, organisms in pond water swimming around and all that kind of stuff. We can look at that under the light microscope. The drawback is that we can only magnify about maybe maximum with the best light microscopes 1,000 to 2,000 times because of this problem, resolution. So resolution is your ability to focus on an image, to see, for example, these two dots that I just drew as two separate dots. And the further back you step, eventually those kind of look like one dot. And you've probably experienced resolution issues with your phone. You try to focus on something, you take a picture, and you start zooming in, but you get to a point where you can't zoom in any further, like everything just becomes pixelated. That's what's going to happen with the light microscope if you go beyond about 1,000 to 2,000 times. Now, electron microscopes don't use light, they use electron beams, and because of the wavelength of how electrons travel, you can actually get much, much more magnification because of the improved resolution. So because we can focus on things that are closer together, we're able to magnify with some of these electron microscopes 100,000 times. So way, way more. And that's allowed us to learn about cell parts that we wouldn't have known about before the development of those two microscopes. Now, the two different electron microscopes, really quick, the difference between them. The scanning electron gives us a 3D image of the surface of objects. The transmission electron microscope lets us see the inside of a cell in more detail. So let me show you a picture with the three. So this is an amoeba, a paramecium, sorry. This is the paramecium with the light microscope. It's alive in this picture. This is the scanning electron microscope. See how you can see all those little surface hairs? So you get that three-dimensional image of the surface. You can kind of see the hairs here, but not very well. And then this is the transmission electron microscope, and this actually allows you to focus on these structures inside that even though you can kind of see them here, if you tried to focus in on these under the light microscope, it would get blurry. So, the cell theory says a couple of things. Number one, all living things are made of cells. It also says they're the smallest unit of life, that they form all the structures and perform all the functions of living things. And then it also says, so all that's sort of encompassed here, and then all cells come from other cells. In other words, cells divide, and they pass on their DNA, and you get new cells from your old cells. Checkpoint question, they want you to match which microscope you'd use to look at some different things. So the first one, the changes in the shape of a living human white blood cell. Well, the living is a clue here. If we go back, remember how the only one that would let us see living things is the light microscope. So this would have to be the light microscope. The finest details on the surface texture of a human hair. So surface, let's go back. Remember, 3D image of surfaces. That's going to be the scanning electron microscope. Sometimes it's called the SEM, scanning electron microscope. And then finally, the detailed structure of an organelle. That's going to be inside of a cell, and so that's going to be the transmission electron microscope. So both of these are electrons. Sometimes this one's called the TEM, transmission electron microscope. Okay. So this figure kind of gives you an idea of why we didn't know about cells until microscopes were invented. With just your eye, notice how you can't really get smaller than maybe a paramecium, and honestly, you wouldn't see it in this amount of detail either. You wouldn't be able to see the hairs on it or anything. Animal and plant cells and bacteria we could see with the light microscope. But then we can't even see the tiniest cell parts like the mitochondrion or the ribosomes. And we definitely couldn't see viruses with our light microscope either. But the electron microscope would allow us to see those things. And notice the measurements here. If you go back to our metric units, micrometers, nanometers, these are very, very, very tiny, tiny amounts. These are millimeters here. So we can see millimeters, but we can't see things that are only a few micrometers in size. And we definitely can't see anything that's only a few nanometers in size. All right, next thing that I want to talk to you about really quickly is that cells need to be small for a reason. You know, why aren't we just made of bigger cells? And the answer is this, the surface to volume ratio. I'm going to show you a really good picture of this on the next slide. In other words, if I just have one giant cell and I need stuff to get to the center, the only way it can get there is from the outside, right? So the more exposed surface I have, the faster stuff can get to the middle. Well, if I have this little bitty cell, the stuff doesn't have to travel as far, right? It can go in at all these surfaces, and it only has to travel a much shorter distance. So a small cell would let things get to the middle faster. Another option that you're going to see in some cells is cells might be long and skinny. Think about that. This cell could still have the same volume, the same amount of liquid inside, but by being this shape, now nutrients and things can get to the middle sooner. The membrane, that's basically what things have to pass through in order to get to the center. It's made out of what are called phospholipids. Now, we talked a little bit about lipids in our last chapter, and this is going to be on the next slide as well. They look like this. They look kind of like a little ball with two sticks. This is the fatty acid part. Remember, fatty acids were the building blocks of lipids. These are hydrophobic. They do not like water. Now, why do I tell you that? Because anytime you want to get something in and out of the cell that's hydrophilic, that likes water, it is not going to be able to go through the lipid bilayer. It's going to be repelled by this whole center lipid portion. Well, then how do we get sugar in our cells? How do we get ions, like our electrolytes, in and out of our cells? And the answer is proteins. So in that cell membrane, you also have channels. They actually look like little tunnels. So you have the cell membrane with the phospholipids, and then you'll have these sort of weird-looking proteins, sort of like this. And these are actually passageways in and out for things that cannot pass through this phospholipid bilayer. The only things that can get through these lipids are hydrophobic things. Oils, steroids, cholesterol, things like that can pass through this bilayer. But anything that's hydrophilic, like water, like sugars, like ions, they cannot. So, a couple of pictures here to show you this. So they want you as a checkpoint to kind of check this and conclude about cell size. So think of this. Think of this as a giant sugar cube, 3 by 3. This is the volume, 27 units to the third. This is the surface area, 54. If I divide that, 54 over 27, I get a ratio of 2 to 1. What does that mean? That means I have 2 times as much exposed surface as the inside. Now let's look at cutting that big cell into all these little cells. Now imagine each of these little cells is completely surrounded by its own membrane. So now all of this is now surface. In between here, there's surface. In between these two cells, there's surface. Well, look how big my surface is now. Still the same amount of contents, but the surface is much more, and now there's 6 times as much exposed surface. I told my class, if you were absent, I said, imagine these were sugar cubes you dropped into water or into hot tea. If I want to dissolve sugar in my hot tea, this and this would be the same exact amount of sugar. They have the same volume, so this is the same amount of sugar. But for this one to dissolve, it can only work, the hot tea can only dissolve it from the outside in. Now I drop all these little tiny balls of sugar in there. This is going to dissolve much faster because each little piece, the hot tea, can start dissolving from the outside of each one of these. So, bottom line, that's why you're made of lots of little cells and not just a few big cells. Your cells need lots of exposed surface for things to get in and out. And when you do have a few of your cells that are bigger, they tend to be longer and skinnier. That way, there's a lot of surface still to get things in and out. Here's a picture of the cell membrane. So again, you have your hydrophilic heads. These are the phosphates. These are made out of phosphate. And then your hydrophobic tails, all this orange, this is made out of fatty acids, which are the building blocks of lipids. All of these are the hydrophobic part. So any kind of ions and sugars and things like that are just going to bounce off the surface. And if you wanted to get something out, it's going to do the same thing. It's going to bounce off the surface on the inside. So to carry any of those things in and out of the cell, we have proteins. Some of them are channels, which just means it's sort of like a tunnel. Some of them require energy. Those are called pumps, those proteins. They may require energy to pump things in and out. We actually have some that are called gated channels that have like a little gate. And only when something stimulates it to open, so it's like a toll road, then stuff can go through. All right, let's talk very quickly. Last thing for today, prokaryotic versus eukaryotic cells. Now, all cells have some cell parts in common. Every cell has a membrane, DNA, ribosomes, and cytoplasm. But bacteria are made of prokaryotic cells, and all the other things, like us, are made of eukaryotic cells. What are the differences? So they all have these things. This is definitely something that will be on a test. Sometimes it'll be a test question. It'll say, which of these is true about prokaryotic cells? And one of the choices will say, they don't have DNA. Not true. They do have DNA, but they don't have a nucleus. So their DNA is not inside a nucleus, it's just outside floating in the cytoplasm. So they still have cytoplasm, they still have DNA. Eukaryotic cells also are bigger. They also have a bunch of organelles, which we're going to start talking about tomorrow. These are all the little tiny cell parts that do all kinds of different jobs. Here's a prokaryotic cell. Here's the chromosome. This is the DNA. No nucleus around it. Has a cell membrane. Most of them also have a cell wall. The nucleoid, is what they call the area where the DNA is. They still have ribosomes, which are these little dots. They still have cytoplasm. And then they may have some other parts that we'll talk probably about later in the year. Eukaryotic cells have all these organelles. Now, what's the benefit of having these organelles? It gives us tiny compartments. It makes the cells compartmentalized. Why is that important? Each of those compartments can be doing a different job. Sort of like in your kitchen. You can heat the oven to 450 degrees without hurting yourself, because the oven, the 450 degrees, stays in the oven. Well, in a eukaryotic cell, you can have a job happening in the nucleus, a different job happening in the mitochondria, a different job happening in the Golgi bodies, an even different job happening in the endoplasmic reticulum. And all these compartments allow the cell to do all kinds of special jobs that a prokaryotic cell just can't do, because it doesn't have all these little compartments. This last thing here is just talking about some of the organelles we're going to be talking about. So nucleus and ribosomes, that have to do with genetics. The organelles that are involved in moving things around, building, breaking, transporting. The organelles involved in energy, mitochondria and chloroplasts, and then the cytoskeleton, which is for support. And plants are different because of these: cell wall,